this morning, having looked at general features of communications in terms of, the raw physiology, I guess, we're going to go on and we're going to look at communication at different levels within the nervous system. We're going to start small and we're going to get bigger. So we're going to look today at communication within individual neurons, how do different parts of a neuron speak to each other, and then look at communication between neurons. And we're going to build up from the first stuff that we did in the first week, where we were looking at action potentials and membrane potentials, to actually look rather than at the nuts and bolts of the electrical activity and how does it move along the axon and so on. We're going to look at how different neurons use that to their advantage to enable them to provide their different functions. So we'll start off with Santiago Ramon y Cajal, another one of the great fathers of neuroscience, winning the Nobel Prize in 1906 jointly with Golgi. I showed you a picture of a Golgi stain of a neuron in the last lecture. This is the kind of diagram that Cajal produced based on Golgi stains of the ways in which neurons interacted with each other. And Cajal became the first person to characterize the cellular structure of organs like the cerebral cortex, which is where this drawing is taken from. The functional superiority of the human brain is linked up with the abundance of the neurons with short axons, the interneurons. And what he hypothesized was that those shorter neurons, the seemingly insignificant neurons, were actually the neurons that were responsible for the great thinking power that many of the primates particularly, but other groups of mammals have compared to the other animals. We often attribute the thinking power that we have to the size of our brains, or the size of our brains relative to the size of our bodies, but that's not necessarily what gives us the ability to think. If I illustrate that, this is a graph of the relative proportion of grey matter and white matter in the brains of a whole host of mammals. And you can see that some mammals have bigger brains, elephants and pilot whales, and some animals are creatures of little brain. Pygmy shrews and common shrews, and Winnie the Pooh is probably down here somewhere, but they're all on that line. So the actual proportion of white matter and grey matter that makes up the brain of a mammal is fairly tightly fixed. It's really difficult to get away from that ratio that goes all the way up from the bottom of the scale to the top of the scale. So if it's not the proportion or the amount of grey matter, the size of our brains, that's important, what else can we look at to find the significant difference between ourselves and some of the other mammals? Well, if we look at the interconnections of central neurons, and for example we compare the connections per pyramidal neuron of a mouse with a human, you can see that each of our neurons has got a huge increase in connectivity compared to those of a mouse, almost 2 orders of magnitude. So what we're saying is, it's not really important how many neurons we've got packed into our skulls within reason. What is important is that each of a mouse's neurons can speak to 3,000 other neurons, and each of our neurons can speak to 100,000 other neurons. And so when we look at neurons, we should be looking for that connectivity. We should be looking to see how they interact with each other and which other neurons they interact with. And that connectivity is the key to function within the central nervous system. Let's go on to look at some of the things that Cajal defined when he was looking at neurons and how that leads into our modern understanding of a neuron. First of all, the doctrine of the neuron. It's a doctrine because it hasn't really been proved to date, and a few odd theories have come along which have contradicted it. But to be honest with you, just about everybody in the scientific community believes that the nerve cell is the structural and functional unit of the central nervous system. People have come along and said, what about those glial cells? There's lots of them. They're really important, obviously, and they're important for some things, but in terms of processing of information, the glial cells don't seem to be particularly important. The other thing that came along with the doctrine of the neurone is the law of dynamic polarization. And that says that excitation can only be transmitted from the axon of one neuron to the dendrites or soma of another neuron. So what we're saying there is that the synapse is a one-way process. I'll show you in a few slides' time that information can go in lots of different directions within a neuron. But once it gets to a synapse, it can only go from the presynaptic side to the postsynaptic side. It can't go the other way. And this has been shown to be not absolutely true, but generally true. In the sense that if you think back to when I was talking about synapses in the second lecture, you'll remember the electrical synapse, the gap junction, which in some cases is bidirectional. But for the most part, these synapses are unidirectional. They're like the valves in the system that stop information going backwards and forwards and route the information down into one particular pathway. A century ago now, the diagrams of Kujal, the doctrine of the neurone, and the law of dynamic polarization led to the concept of the generalized neurone. And this is the sort of schoolboy neuron that we all meet when we go to high school and we all draw and we all think about as a neuron. It has specific excitants at one end. We know those as neurotransmitters. These excitants lead to a graded electrogenesis, a graded electrical change in membrane potential. And we now know that that's through inhibitory and excitatory postsynaptic potentials. At some point, this graded electrogenesis gets turned into a point of information or an action potential which is transmitted to the other side of the neuron or the other end of the neuron, and that becomes secretory. And so when we think about neurons, in terms of drawing diagrams with 10 or 20 different neurons on, we tend to draw lines and very simplified diagrams, but what we're drawing is groups of generalized neurons. That's kind of what we're thinking about. The other thing to say about the generalized neuron is that it's only really interested in the electrical function. We know that neurons have complex metabolic functions and that those metabolic functions are essential for information transfer. And we know, and you'll find out in the lectures towards the end of the program, that different neurological diseases cause us to be less able to process information because of changes in metabolic function, which kind of unhook this information process. But in the healthy, normal brain, when we're thinking about information processing, we're only thinking about electrical function. And that kind of fits in with this doctrine of the neurone, that the neurone is effectively an electrical processor of information. So here is that schoolboy neuron. It's a lower motor neuron, so it has a cell body at this end, which is taking in information from specific excitants, transferring it down an axon to a block of effector tissue, in this case skeletal muscle. And so that's the kind of neuron that we all think of, or most of us think of, when somebody says, what does a nerve look like? That's the thing that you're most likely to draw. But there are lots of different nerves, and so I just want to go over some of them now and talk about how we classify different nerves and how we grade them. We've got the general neuron, and here we've got the schoolboy lower motor neuron. But you can see here that we've got lots of other models of neurons, all of which have different sort of schematics based on that general neuron. And some of them are a little bit odd, like, for example, the cutaneous sensory receptor, which has its cell body on a stalk in the dorsal root ganglion and kind of has, well, what does it have here? We call these dendrites and we call all this axon. But if you think about it in terms of the concept of the generalized neuron, we should probably call it dendrite down to here and axon down to here, but it's all the same. And so we tend to call dendrites and axons together neurites. And a neurite is a generalized term for an extension of a nerve cell, whether it be a dendrite or an axon. And it doesn't really matter whether it's a dendrite or an axon, because they tend to do the same sort of thing. But we would tend to call these things in a proliferating tree up here dendrites, and this long extension down here an axon, both neurites. This kind of picture of a generalized neuron tends to break down even more when we start considering the short neurons, the interneurons, those important connectivity neurons. And if you can see when we get down here to the retinal amacrine cell, it's a very strange neuron. All its processes are in one direction. It has this cell body that doesn't really seem to be very involved in things, and it doesn't undergo action potentials. It processes information solely on membrane potentials. So the point I'm trying to make here is that the generalized neuron is a good way to think of neurons in terms of function, but the actual structure of those neurons sometimes stretches it and pulls it to the point where it's no longer really a very sensible way to model a neuron, and we even make up new language to describe some of those neurons because they don't fit with what we know as the generalized concept. But in terms of function, the generalized concept works well for most neurons most of the time. So it's a good model to look at as exactly what it is, a generalized neuron. When we classify neurons, we start off with unipolar cells. They have one process that comes out from the cell body, which is an axon, and that kind of becomes dendrites and becomes a dendritic tree. It's a unipolar neuron, one pole, one process. And we don't see those in vertebrate anatomy. They are quite common as invertebrate neurons, but the vertebrates don't seem to have these very much. The next kind of cell that we see is the bipolar cell. And here the cell body has got two processes tending to go in different directions. And so now this is looking more like the classical thing, and we're tending to call one process dendrites and the other process the axon, although that's kind of flexible. And the example there is the bipolar cell of the retina. This is a sensory cutaneous neuron. So its dendritic tree is in the skin of wherever it's innervating, and its axonal processes are in the central nervous system. And you can see that it looks a lot like this bipolar cell. Yeah, it's got processes going in two directions, but actually it's only got one process that leaves the cell body, because the cell body is tucked away in the dorsal root ganglion. And so it's kind of unipolar and bipolar together, and we call it pseudo-unipolar, because it's kind of a bipolar cell functionally, but it's pretending to be a unipolar cell. So it's pseudo-unipolar. And these are the longest neurons in the mammalian body. This end can be in your, I don't know, left big toe, or right big toe, or wherever. This end can project all the way to your brainstem. So these things can be nearly 2 meters long in people, and in larger mammals like horses up to about 3 meters long, a single cell. They are the longest of the neurons. When we get into the central nervous system, we start to get more diverse classifications going on. And we talk about multipolar neurons. So once a neuron has more than 2 projections from its soma, we call it multipolar. And we don't really worry about how many it's got. Examples: the lower motor neuron of the spinal cord. I showed you the Golgi stain in the last lecture. The pyramidal cell of the hippocampus has this characteristic structure that looks, well, I'm not entirely sure what it looks like, but it is characteristic. So it's got these apical dendrites extending into the superficial layers of the hippocampus. It's got these basal dendrites which kind of extend around in a fan around it. And it's got axonal processes, some of which loop back and go up into the apical dendrite tree. But it's once you've seen that a few times, you'll instantly recognize it as a pyramidal cell of the hippocampus. There's not really very much else that looks like that. And another example here, the Purkinje cell in the cerebellum, with a long axon, a cell body, and this really intricate dendritic tree. Just something to mention about this, we'll talk about it again when we talk about the cerebellum later on, but this dendritic tree of the Purkinje cell has a particular shape in that it's almost 2-dimensional. It looks really sort of wide in this plane, but if I took this diagram out and turned it through 90 degrees, it would be completely flat. It's a 2-dimensional processing. It goes wide and it goes tall, but it doesn't extend into this plane. And when we look at different Purkinje cells in the cerebellum, they line up against each other like slices of toast in a sliced bread. They're all next to each other. They don't interact with each other's dendritic trees. So not all neurons follow the general pattern of structure, and we deal with that with those classifications. Not all neurons follow the general pattern in terms of function. Some of the examples I want to talk about: action potentials in dendrites, in dendritic trees; dendro-dendritic interactions between the dendrites of 2 neurons; and neurons that don't have the ability to generate action potentials. We all know that let's use the example of a lower motor neuron. The action potential starts at the axonal hillock and propagates down the axon to get to the synaptic boutons or knobs and to generate chemical information which crosses to the next neuron, or to the muscle fibre in this case. So the action potential travels along the axon. If you were to take one of these axons and stimulate it electrically halfway along, which direction would the action potential travel in? Both. Yeah, that's right. It goes in both directions. There's nothing in this axon which confers any directionality on the action potential. So if we start an action potential here, then the action potential goes both ways. And that's also true of the axonal hillock. The action potential starts at the axonal hillock and travels along the axon, but it also back propagates and travels along the soma of the neuron and even through the dendritic tree. And this back propagation, although it can't cross the synapses backwards, it kind of resets the balance of EPSPs and IPSPs within the neuron and kind of resets the neuronal memory. The back propagation isn't always complete, and I'm about to demonstrate that with the next 2 slides. If we inject current into a neuron, either with an electrode or through excitatory postsynaptic potentials, we know that we increase the rate of the action potentials that we generate. And if the back propagation was complete along the dendritic tree and throughout the neuron, we would get a single curve described on this graph, so that greater injection of current would always result in a steady increase in the rate of impulse generation. But what we actually see is that there are several processes going on here. And here you can see that there are two parts of this curve, both with different rate constants. And this indicates that there is some memory going on within the neuron for the situation prior to the generation of its last action potential, and that the back propagation doesn't completely wipe the slate clean and wipe out the neuron's memory. Action potentials can originate and travel through dendritic trees. We tend to think of the dendritic processes as conveying information by changes in membrane potential through inhibitory and excitatory postsynaptic potentials. But you can see from this recording made in the dendritic tree of this neuron that there are action potentials at different points in the dendritic tree. So that if you look at the electrical activity at the cell body, you can see that we're getting these nice bursts of action potentials. When you look at the action potential activity throughout the dendritic processes at different points, you can see that there is different activity at different points, which is contributing to these nice bursts that we're seeing in the soma of the neuron, which is being propagated down the axon. It's also worth saying at this point that information that neurons convey is not just conveyed as single action potentials, but it's conveyed as bursts of action potentials, and the duration, the rate of action potentials within a burst, and also the pattern of bursting can be important ways in which information is coded. If we look at the effect of action potentials in dendritic trees, then we can see that they can have quite a profound effect on the nature of discharge of the neuron. This is a hippocampal neuron, and you can see at the top that it's got a very arborized dendritic tree with a circular area central to the neuron and also a distant area where there are dendritic processes. If you look at the pattern of firing of this neuron, when all of the dendritic processes are active, you can see we get this pattern at the top where we get regular bursts of multiple action potentials. But if we inhibit the arborized part of the dendritic tree and leave the neuron just working on its localized area, you can see that the pattern of discharges in the body of the neuron changes considerably, so that we are getting more rapid action potentials but single bursts of action potentials. So the action potential bursts are coming more rapidly, but they contain fewer action potentials. So you can see how synapses and inhibition at a particular point in the dendritic tree of a neuron can have profound effects upon the discharge pattern of that particular neuron. We also need to consider dendrodendritic interactions. Classically, we've considered synaptic interactions as being interactions between the axons of one neuron and the dendrites of another neuron. But you can see here in this diagram of the sense of smell that there are interactions here, synaptic interactions between dendrites of mitral or tufted cells and dendrites of granule cells. And these dendrodendritic interactions are extremely complex and involve dendritic processes of one neuron influencing the activity of the dendritic processes of another neuron. This is a diagram of what we know about the molecular biology of these dendrodendritic interactions, and you can see that they are indeed extremely complicated. There are 4 different sorts of projections coming into these neurons, and you can see that we've got AMPA receptors, NMDA receptors, serotonin and 5-HT3 receptors, GABA-A receptors, dopamine release and dopamine receptors, all contributing to the activity at these interactions. So they are extremely complex information processing units, and they are affecting information processing between dendrites of one cell and dendrites of another cell. Some neurons process information without generating action potentials. This is a diagram of part of the retina, and you can see that we've got the signal transducers along the top, the rods and cones, and they respond to light striking their cells by altering their membrane potential. And as information passes through the retina, the bipolar cells, the horizontal cells and the amacrine cells respond to stimulation by altering their membrane potential. They are incapable of generating action potentials. The first cells in this pathway which generate action potential are the ganglion cells down at the bottom here. And these are the cells whose axons contribute to the optic nerve. But they are the first cells that generate action potentials, and the membrane potentials of the cells which occur prior to them in the information processing pathway alter the rate of generation of action potential in the ganglion cell. So information can be processed as action potentials, but it can also be processed as changing membrane potentials. We saw before the profound effects that inhibitory neurons can have synapsing on a particular part of the dendritic processes of a neuron. And what we need in these places and throughout the dendritic tree is a mechanism by which different neurons which affect the tree at that point can summate their activity so that the information is processed in an appropriate manner. If just one neuron synapsing on that dendritic tree there could have that profound effect on this particular neuron, then that might reduce the subtlety of information processing, and so we need ways to summate the information at these different locations. The way in which this is done is through dendritic spines. These are processes actually on the dendrites which act to allow information to be summated. You can see here in this stained section that there are bobbly processes all the way along the dendrites, and these are the so-called dendritic spines. You can also see in the bottom picture more detail of a particular dendrite. You can see that individual spines have varied shapes and sizes and thicknesses and knobs on the end of them. So you can see that these dendritic spines are different shapes and sizes, different sizes of buttons on the end. And the question is, how do they augment the processing of information? At first, they were thought just to increase the surface area for synaptic attachment. But then it became apparent that they may electrically isolate areas of dendrite or biochemically isolate areas of dendrite, and also that they may be involved with learning. What appears to be happening is that as learning takes place, the dendritic spines can change their shape. And by changing the length and the diameter of the pedicle that they attach to the dendrite with, they can alter the resistance between the actual knob at the end of the dendritic spine and the dendrite itself. This means that the activity in the dendritic spine will have more or less influence over the electrical activity in the dendrite. So that a long, thin process with a dendritic spine at the end will have a high resistance between itself and the actual shaft of the dendrite, and so will only have a small influence on the dendrite, but a shorter, fatter peduncle will have much less resistance, and so will be much more easily able to affect the electrical activity within the dendrite. It's also apparent that different groups of synapses attach onto different areas of the receptive field of a neuron. So here we have a secondary sensory neuron with its cell body in the spinal cord and its dendritic tree indicating the area of spinal cord which it's gathering information from.
on the right, we have the area of the animal's paw, which is the sensory field of this neuron. And you can see that if we stimulate a point in the centre of that sensory field, then the points on the dendritic tree which are stimulated on this neuron are highlighted in red and tend to be central. They tend to be close to the soma of the neuron. Conversely, if we stimulate peripherally on the sensory field, we tend to get fewer points of stimulation, and they also seem to be further away from the body of the neuron on the edges of the dendritic processes. And so what we're finding here is that as we stimulate neurons, they are more receptive to stimulation in the centre of their receptive field compared to the periphery, and we can also change the degree to which they're stimulated by changing the shape of the dendritic processes. You can see that even more if you look at the receptive fields of hippocampal neurons or any large central nervous system neurons. You can see in this hippocampal neuron that different classes of fibres which have synapses with this pyramidal cell tend to interact with it in different places. So that, for example, the body of the neuron tends to have synapses from the endings of basket fibres, whereas the peripheral apical dendrites tend to receive synapses from the endings of short axons and afferent fibres. In the area slightly lower down, there are Schaefer collaterals and then mossy fibres, and then down on the basal dendrites we tend to have collaterals from other pyramids and more short axons. So the area onto which a synapse interacts with a dendritic tree can affect the action of that synapse upon the cell.